Hello everyone. First of all, I would like to thank the organizers for the invitation. I truly appreciate it, especially because this is a very special moment for me and for all the people involved in directed molecular evolution. It's just amazing that eventually Frances has achieved such a high recognition, which she truly deserves. And my talk for today is, of course, about directed evolution. And I've divided the presentation into sections. First of all, a small introduction about the history of directed evolution and some general concepts about the technique. In case you are not familiar with directed evolution, I hope that at the end of this talk at least you understand the general picture of a directed evolution experiment. And I'm going to illustrate the directed evolution with some case studies taken from my own laboratory because as you can understand, I'm really familiar with my own research. To make this more interactive, if you feel or if you find questions, just interrupt me and we can discuss during the talk. Okay? So let's start from the beginning, the history of directed evolution. Well, let me first tell you that something about enzymes. Enzymes are proteins with catalytic activity. They are wonderful biomolecules that can speed up chemical reactions in nature up to the order of 10 to the power of 19 folds. They can work in an environmental friendly conditions in terms of they work in water at room temperature, under atmospheric pressure. They are highly non-stereo and selective molecules, and they release very few side products. However, when we take out an enzyme from its natural environment and we put the enzyme within a given industrial context, the enzymes quickly inactivate it. Why? Basically because enzymes are used to work in such a benign environment and regulating strictly metabolic pathways. And when we take out the enzymes from this environment and we put in a harsh condition, the enzyme is not used to work is not ready to work under such harsh conditions. And they inactivate it, and therefore, unfortunately, they don't work as we expected. And that's why protein engineering arose many years ago. To meet enzymes with industrial standards in terms of improve the properties of enzymes versus solvent tolerance, thermostability, or pH range, to alter specific properties in terms of enhance the broad substrate range, the stereoselectivity to reduce the inhibition against different types of inhibitors, or to improve expression levels in heterologous host from scratch. Also to understand how enzymes can work in terms of making structure-function relationship studies. And last but not least, to discern fundamental questions behind natural protein evolution, which is also very interesting from a scientific point of view. And this story actually started with Kary Mullis. I don't know whether you know this guy. He was a scientist who was working in the early 80s in Southern California in a small company called Cetus. And he invented the PCR reaction, the polymerase chain reaction, by which, for the very first time, we are capable to amplify tiny amounts of DNA with high fidelity to obtain considerable amounts of DNA. And this was a revolution in terms of protein engineering, because for the very first time, we are capable to manipulate the proteins at the genetic level. The story of this guy is very interesting because Cetus gave him as a prize for his discovery a check of $10,000, and afterwards they sold the patent to Roche. The company Cetus sold the patent to Roche, a big pharma company, for $300 million. So you can imagine the face of this guy when he realized about this, but eventually he was paid it off because he got the Nobel Prize for this invention in chemistry in 1993. And afterwards he spent most of his life making surfing because he's from Southern California and he loves surf. And in terms of the impact of the polymerase chain reaction, well, changed many things in our lives. For example, when we are really familiar now with paternity tests, but believe me when I say that in the early 80s, no one could make this kind of a test. Now is the son of that Vader because of the movie, and also because we have paternity tests. And also it's important, it has a strong impact in forensic medicine, to give you a couple of social applications of the PCR. But in terms of protein engineering, was the first step towards a wonderland of rational design, which is so-called site-directed mutagenesis. The goal of a site-directed mutagenesis experiment is to design an enzyme whose crystal structure is available through point mutations in specific residues. So, first of all, we need the crystal structure of the enzyme. And we can speculate about this crystal structure. We can somehow try to change several residues by site-directed mutagenesis in order to modify the properties of the enzyme. So we take a look at the crystal structure and we decide, okay, we are going to change this residue because we believe that this valine at this position somehow could affect the kinetic properties. Yeah?
Sorry, I'm a bit confused when you speak of crystal structure here. Do you mean the way the atoms are ordered within the protein? What's the three-dimensional arrangement of the protein? Or
exactly.
Different proteins, I mean,
no,
different individuals of the same protein arranged in space?
No, the crystallization of enzymes, basically, or of protein in general terms, is to get the 3D dimensional vision of the whole enzyme. And to do this, you have to crystallize the protein. You have to produce the protein, purify the protein, and afterward get the crystals of that protein. And you have the crystals in different types of resolution, and in this way you have a general overview of the structural organization of the protein. This is what we call about crystal structure. And of course, all the atoms, how they are organized within the network of the whole three-dimensional structure of the protein. This is a crystal of a protein. Okay? So, basically, once we have the crystal, so this is one of the main bottlenecks of site-directed mutagenesis. To make site-directed mutagenesis, you need the crystal structure of the protein, and this is not so straightforwardly accomplished. It's really difficult to get crystal structure with high resolution, with all the atoms correctly ordered, and besides, once we speculate about which changes we could make here, we can go to a genetic level, to the gene which is going to codify for this protein, and make, by manipulating the PCR reaction, a change, which basically means a mutation at genetic level, at the codon, giving rise to a new amino acid at a given position. Afterwards, we introduce this gene within an heterologous host, we express the enzyme, and if everything goes right, we are going to have an enzyme with a change in the position we already mapped in our crystal structure, and afterwards we evaluate the biochemical properties of this enzyme in terms of kinetics, thermostability, etc., to see whether or not the changes somehow are related to the new properties of the enzyme. This is the so-called site-directed mutagenesis or rational design, because we anticipate, we speculate about which changes we're going to make, and afterwards we have to make these changes and to see whether or not these changes are successful in terms of the property we're going to improve or study. But the main bottlenecks about rational design are basically two. First of all, we have low availability of protein structures. Even though we have a revolution in terms of genome databases, etc., it's very difficult still to get good crystal structure of enzymes. Therefore, we are limited by the crystal structure. And second, and more importantly, we have to consider enzymes, or proteins in general terms, as flexible molecules. They are actually really flexible and dynamic molecules, which somehow are showing different conformational changes at local but also at global level with the entrance of the substrate and with the release of byproducts. And this is something that is not taken when you are taking a look at the crystal structure of an enzyme. Somehow the crystal structure of an enzyme is just a moment in the life, in the kinetic life of the enzyme, and unfortunately we are far away of understanding how the kinetics and the behavior of enzymes is truly working. So it's very frustrating to make a lot of speculations, a lot of hypotheses about how an enzyme is going to work, make the directed evolution, make the site-directed mutagenesis experiment, and afterwards see that actually it's not working as you expected. And to solve this problem, in the early 90s, Frances Arnold from the California Institute of Technology, my mentor in directed evolution, came with the idea of taking advantage of the power of natural evolution. And she invented this wonderful and revolutionary technique, so-called directed evolution, by which she just been granted with the, she has been awarded with the Nobel Prize. And I've done many interviews in the last few weeks because of this Nobel Prize, because people knew that I was working with her. And it's curious that in some channels they ask me whether or not she was awarded because she was a woman. And definitely this award is really deserved. If you take a look to the trajectory of Frances Arnold, she was awarded first in the 2011 with the Draper Prize, which is the equivalent to the Nobel Prize in Engineering, along with Pina Stemmer, which unfortunately passed away many years ago. Afterwards, she was awarded again with the National Science Technological Medal in 2011, and very recently with the Millennium Technology Prize, which is the equivalent of the Nobel Prize in Technology. And besides, she belongs to the three National Academy of United States of Science, Engineering, and Medicine. She's the only woman who belong to these three different National Academies of Science. So definitely, she truly deserves this Nobel Prize for this invention. It's not because she's a woman. I want to point out this here because it's curious that people ask me these kind of questions. So what I was saying was that, yes, actually Frances Arnold has done in the last couple of decades an extraordinary effort trying to transmit the significance of her research and to create a school of protein evolvers all around the world. And among her most important successful case studies are, for example, the production of biofuels, the synthesis of chemicals. She even has founded a couple of companies. One of these is Gibo, which is leading now the production of isobutanol in the United States. And very recently, with other students from her group, she founded Provivi, which is trying to replace pesticides using strategies of directed evolution. And in terms of science, she's and that's why I think she was granted she was awarded with the Nobel Prize. Especially in the last few years, she has focused her research on non-natural chemistry. She's trying to manipulate enzymes to make chemistry which is not naturally occurring in nature. For example, to prepare molecules that show carbon silicon bonds, or to prepare molecules which have carbon boron bonds for different materials for applications in medicine, etc. And if you are wondering what is directed evolution, now I'm going to explain to you. So directed evolution basically is a wonderful strategy to tailor enzymes, metabolic pathways, or even whole microorganisms with improved traits. Through iterative steps of random mutagenesis, DNA recombination, and screening or selection, and by mimicking the Darwinian algorithm of natural selection, we can reduce the temporal scale of evolution from millions of years until I would say only days of work on the bench of the laboratory. While in natural evolution everything happens over millions of years, we are reducing the temporal scale of evolution. And besides, in natural evolution everything is happening spontaneously, whereas in directed evolution the selective pressure is strictly controlled by the scientist. And this is the classical directed evolution round, where we start either using one single enzyme, one single parental type, one single gene which is going to codify for our desired enzymes, or with a collection of different enzymes which share a certain degree of sequence identity between each other. And by molecular biology techniques, we introduce randomly mutations in the genes, or we recombine the genes, as also happens in nature during the sexual approaches. Afterwards, these mutant libraries are going to be inserted within a suitable heterologous host to induce the expression of these proteins, the expression of these enzymes. We express the enzyme in such a way that in every single clone we are going to have a gene which is containing a recombination event or a mutation which is going to give an exclusive property to the enzyme which is codified by that gene. And the clones are growth in this kind of format, which is so-called high-throughput screening format. And we analyze every single clone by high-throughput screening technologies. I will show you in a couple of slides how we do this. in such a way that we select the winners, the best performers of this first round of evolution against a selective criteria. Let's say that we want to improve the thermostability of an enzyme. We incubate these plates at high temperature. We want to have enzymes that somehow recognize a new substrate. We offer to the mutant library the new substrate. Only those variants who are somehow recognizing the substrate and giving a colorimetric or fluorimetric response are selected as parental types for a new round of directed evolution. And we repeat this process many times as necessary, until we improve notably the desired trait. So basically directed evolution is mimicking the Darwinian algorithm of mutation, recombination, and selection, so that we can enhance different properties of enzymes in terms of activities. We can create novel substrate specificities, we can modify radiochemia and stereoselectivity, we can create substrate promiscuity, catalytic promiscuity, and in terms of stability, we can improve enzymes at high temperatures in the presence of organic solvents against different types of inhibitors, or just to enhance the functional expression levels in different heterologous hosts. The final goal is the design of customized biocatalyst. And this slide is summarizing some of the news which came out many years ago in the early 2000s. And at the very beginning, the people who were a little bit reluctant about directed evolution. I remember very well when I returned to Madrid from Caltech, and when I said to some of my colleagues that I was planning to work on directed evolution, the people was reluctant because this is something new, and sometimes we don't truly understand the impact of this kind of research. But fortunately, at the end, the research succeeded, and it has done a big impact in society. This is an example taken from some years ago about the application of directed evolution, for example, the biomedical application in the production of or in the application in blood banks. A consortium of researchers from California and also from hospitals in Southern California creating an enzymes that somehow were capable of treat the human blood in order to transform the different types of human blood, A, B, AB, into the universal donor, into the type 0. And this is on a slide where you can see the growth in research papers of directed evolution, and also directed evolution evolved by the hand of the industry. All the biotechnological companies are setting up different research lines and different divisions for a directed evolution of their enzymes. For example, Novozymes, which is the global leader company in industrial enzymes, if you take a look at its catalog, you're going to have enzymes for laundry, for baking, for biofuels, for animal feed, for textile and leather, etc. Believe me when I say that most of these enzymes have been manipulated in one way or another by means of directed evolution. Also, we can see here Codexis. Codexis is a strong company which is also focusing its research, for example, in the synthesis of pharma compounds like Januvia for the treatment of diabetes, or in the evolution of carbonic anhydrases for the capture and sequestration of CO2. All these examples are successful case studies about directed evolution, which are right now in the market. This is Berenin, another company which is also involved in directed evolution, and they are evolving enzymes from extremophile environments. So they believe that if they take enzymes from extremophile environments, let's say that they explore a volcano and they take samples from there, they believe that these enzymes are going to be better departure points for directed evolution because they are more robust enzymes, and therefore you can improve even the robustness of this enzyme further. And this is just a kind of exotic case, the Genecore, which is creating cocktails in case we are in a chemical warfare defense. So basically, directed evolution is somehow rewriting the code of life, as Francis says, and actually we are witnessing the big impact that directed evolution is making in our society right now. And now I'm going to explain to you a little bit about the tools that we need to make a directed evolution experiment. First, we need a reliable high-throughput screening protocol to detect the improved variants or traits. Second, we need the functional expression of the mutant library. We need to have in hand heterologous hosts for functional enzyme expression. And last but not least, we also need a range of methods for the generation of DNA diversity. In terms of a high-throughput screening assay, basically means the simultaneous assessment of the properties of the enzyme in a rapid way. And this basically needs an assay that must be robust, reliable, sensitive, and reproducible. There are many ways of making high-throughput screening assays, either in solid phase format, or most commonly colorimetric, fluorimetric assays. We can even combine different assays within the same directed evolution experiment. Let's say that we're going to improve a couple of different properties, the stability and the activity of the enzyme. We create the mutant library, and we screen at the same time for activity but also for stability. We fish the winners, and we recombine those winners in a new round of directed evolution. And of course, we can do this right now with ultra high throughput screening assay coupled to FACS and this kind of systems. As I couldn't take all of you to my laboratory, I brought here a couple of movies about how a high-throughput screening platform works. Here you can see a colony picker, which basically is saving a lot of time. By picking colonies, every single colony is containing a gene which is going to codify for a different enzyme, because you have there the mutations already introduced by molecular biology techniques. And we transfer these colonies to this kind of plates, 96 well plates typically, where we induce the expression of the enzyme. We make the fermentation in this tiny format. I was showing to you the different screening platforms typically used in directed evolution: the colony pickers, the pipetting robots, the liquid handlers, and the plate readers. So you have to have in hand this kind of equipment to make directed evolution experiments, and that's why it's not easily accomplished in many academic laboratories, because these kind of facilities are very, very expensive. And yeah, in terms of methods for the generation of DNA diversity, there are two tendencies. First, what we call the asexual approach, I mean the random introduction of mutations throughout the protein, and the second one, the sexual one, which basically means the recombination of different parental types. To make the asexual approach, the random mutagenesis is mostly, in most of the directed evolution experiments, based on the modification of the PCR reaction. Basically, the PCR reaction allows us to amplify with high fidelity DNA. In this case, we amplify the DNA, but we make the polymerase to make mistakes during the amplification of the DNA. And these mistakes basically means mutations. So we can control the frequency of mutational loading, the frequency of mutations we are introducing, but not the type of mutation we are going to introduce. And the second approach, the sexual one, you are going to let me establish an analogy between animal breeding and molecular breeding. In animal breeding, we use only two parental types. In molecular breeding, we use one, two, or many different parental types. In animal breeding, we only obtain one of a spring. In molecular breeding, we obtain several of a springs. The limitation is just imposed by the screening assay. Here we have strict phylogenetic barriers. We cannot cross a wave with a hang. And here we have a huge flexibility. Actually, we can create really different chimeric proteins with sequence identity lower than 50% in some of the cases. And the temporal cycles in animal breeding are slow, and here we are as fast as the student is working in the laboratory. We can, yes?
Yes, and the kind of mutations that you use are completely random, or you also control the mutations?
They are random. They are random. This is the nice thing about, I forgot to say that when I, at the beginning of my talk, when I was talking about site-directed mutagenesis, the rational approach, you are always planning which position you are going to introduce. In directed evolution, we don't care about that. We introduce randomly everything, we look for the winners, and afterwards, we can, once we have a product, an enzyme that is working, we can speculate about why those mutations gave us these effects. And sometimes we cannot find really or truly understandable explanation about it, but the truth is that the enzyme is working with those mutations. Okay? Nevertheless, right now in directed evolution, people are combining all the different approaches, as you can imagine. Sometimes we combine also rational. Even in my own laboratory, we made a little bit of rational design, but always on the base on directed evolution. We can, for example, find out some mutations randomly, but afterwards we can explore those hot spot residues by rational means. In terms of the sexual approach, I've taken this here because in my laboratory we are really specializing working with Saccharomyces cerevisiae as heterologous host for directed evolution, because we are working with eukaryotic proteins, and when one is doing directed evolution with eukaryotic genes, it's much faster to work in Saccharomyces cerevisiae. And I'm not going to give you the technical details about this, but let me tell you something about Saccharomyces cerevisiae. It has a high frequency of homologous DNA recombination, and this is extremely useful to make methods for the generation of DNA diversity. We can take advantage of this. We can harness the molecular device of Saccharomyces cerevisiae and dress up our favorite host with the custom mechanics to create methods for the generation of DNA diversity. And even though this is something that somehow is out of the scope of my talk for today, please take into account that most of the directed evolution experiments done in my laboratory are supported by the Saccharomyces cerevisiae machinery. And now I'm going to give you, I've chosen 3 case studies to illustrate the directed evolution based on our own research. First, fungal LACCASES in human blood. My laboratory has spent the last 15 years working with the ligninolytic enzyme consortium. This is a wonderful system. Actually, it's considered by many as one of the most efficient oxidative systems found in nature. This consortium of enzymes is secreted by white rot fungi, which are involved in the degradation of wood in nature, in the degradation of lignin. In particular, lignin is this recalcitrant biopolymer which gives consistency to the plant cell wall, while protecting the plant against microbial attack, and also is imbricating between hemicellulose and cellulose fibers. And this consortium of enzymes is capable of degrading lignin even though it's recalcitrant. This consortium of enzymes is formed by high redox potential oxidoreductases, also known as ligninases. Among them, we are going to find LACCASES, peroxidases, peroxygenases, hydrogen peroxide supplying enzymes, and all together are going to interact with each other through a complex cascade mechanism of action which is not fully understood yet. But what we do know is that they are going to find a lot of applications in different biotechnological sectors because of their broad substrate range and their high redox potential. Indeed, we can use this kind of enzymes after making a little bit of improvement by directed evolution in pulp biobleaching, in bioremediation, in biofuels production, in biomedical devices, in organic synthesis, to name just a few examples. And among these enzymes, we are going to find a blue multi-copper containing enzymes which are so-called LACCASES. LACCASEs are polyphenol oxidases capable of oxidizing phenols, polyphenols, anilines, and many other aromatic compounds with a concomitant reduction of molecular oxygen to water. Actually, they are considered by many as the ideal green catalyst because they use oxygen from air, they oxidize a bunch of different compounds, and they release only water as the only byproduct. And let me introduce my friend Sergei Sliv. He's a Russian guy who is working in the University of Malmö in Sweden for many years now. And some years ago, he suggested me to make directed evolution of LACCASES to be active in human blood within a European consortium. And this project, as I've said to some of my colleagues, took us sweat, tears, and blood. I will explain later why about this. So the idea of a LACCASE in human blood is interesting because high redox potential LACCASEs belong to the exclusive group of oxidoreductases that can accept electrons directly from a cathodic compartment. So that if we attach the LACCASE, if we mobilize the LACCASE in the cathodic compartment of a biofuel cell, we can power the whole device. And here we have a biosensor for the detection of different metabolites in human beings. So the final goal of this project was actually to create a 3D nanobio device for the detection of different metabolites in human beings. But the question which is arising here is whether a LACCASE can work in human blood, because LACCASES are used to work in a forest secreted by white rot fungi in a strictly acid environment, and the human blood conditions are very different. Indeed, LACCASES, high redox potential LACCASES, are neither active nor stable in human blood for two reasons. First, because unless you are dead, the human blood pH is around 7.4, where high redox potential LACCASES are fully inactivated. And second, because the human blood composition contains a strong concentration of sodium chloride, around 100-140 millimolar, and high redox potential LACCASES are strongly inhibited by modest concentrations of halides. At this point, we couldn't use real human blood in our screening assay because otherwise a lot of interferences would occur in terms of coagulating agents and red cells. Instead, what we did was to prepare a surrogate blood, which we call blood buffer. And this blood buffer basically mimics the biochemical composition of human blood, but in the absence of cells and coagulating agents, and incorporating a classical LACCASE substrate as reporter of the LACCASE activity. And with this screening assay in hand, we started to make rounds of directed evolution and screening by introducing randomly mutations in the gene, by recombining different genes, using different techniques of molecular evolution, and enhancing the selective pressure from pH 6.5, where the LACCASE activity was hardly detected, until pH 7.4, the pH of human blood. At the end of this project, we found out an enzyme, a mutant, which showed activity at this pH and under these conditions in this surrogate blood. And I immediately emailed Sergei Sliv, and I told him, I have the enzyme, but I don't know actually if this enzyme is working in real physiological fluids, because we've done all the experiments in this surrogate blood. And he told me, Miguel, send me right away the mutant, because I have here the physiological fluids in my hospital, in my university. And we produced the enzyme, and we sent to him the enzyme, and he made the experiments. And indeed, well, this is important to say that we enhance the selective pressure. The gradual increase in the selective pressure, at the beginning the enzyme was not capable to tolerate the pH of the surrogate blood, but at the end of the process the enzyme behaved in a good shape in this pH because of the increase of gradual selective pressure. This is something commonly done in directed evolution experiments, to increase little by little the selective pressure. And indeed, as I've already said, Sergei made these experiments with real plasma and blood, and the LACCASE showed activity in both physiological fluids. When we made a breakdown in the biochemical properties of the enzyme, we realized that the pH activity profile of our mutant not only shifted towards the alkaline side of the pH scale, as you can see here, and more significantly, the tolerance versus high concentration of halides was strongly improved. Afterwards, the LACCASE was included in this 3 nanobio device, the first prototype of this kind of devices for the detection of metabolites in human beings, and we celebrated. And now I'm going to tell you why this project took us blood, sweat, and tears. When Sergei told me, Miguel, send me the sample for the trials here in Sweden, actually he didn't have human blood because there was a lot of bureaucracy behind that, but instead what he did was taking his own blood from his body, and this experiment was done with the blood of Sergei Sliv. So if you take a look at the paper in the supplemental material, it's described that the blood belongs to a Caucasian guy of 40 years old, etc. This is Sergei Sliv. And even better, it's also taking tears because he was so excited with the experiments that somehow he wanted to check out whether the enzyme could work in the eye for the design of lens for the army. Well, this guy is a little bit crazy, and he didn't have tears, so with an onion, he made cry one of his students, and with a capillary, he took the tears and made this experiment. Well, this is the price of science. Anyways, and we've done a lot of experiments with LACCASES. I've taken this one because it was recently developed in my laboratory, and it's also because Francis Arnold was behind this somehow, and it's the creation of chimeric enzymes. I mean, this is not possible in nature, right? We cannot create these strange animals, but what do achieve by directed evolution is chimeric proteins, and actually in collaboration with Francis Arnold, who developed this wonderful method which is called schema. Schema is an algorithm, a computational algorithm to predict in silico which kind of recombination events you should make in a directed evolution experiment to create chimeras with high functionality. And we applied this algorithm in collaboration with Francis Arnold to create chimeras of different types of LACCASES. And we obtained a collection of different chimeras which improved notably their thermostability, and also they showed hybrid properties in terms of substrate range. And a second case study, I've chosen the resurrection and evolution of ancestral enzymes, something in which we are deeply involved right now. To understand what enzyme resurrection is, we have to come back to the basics. All of you know that primitive cells contain only a small set of enzymes because they couldn't afford, from a metabolic point of view, to have a huge amount of different enzymes specialized in every single aspect of the metabolism of the enzyme. And in the course of natural evolution, this generalist biocatalysts, because actually they display different activities to withstand the survival of the host, they became specialists, losing most of these activities which exist many years ago in the Precambrian era, for example, whereas enhancing other activities. And these are the activities that we have right now in our modern enzymes, in our planet right now. There is a revolution in genomic databases and in protein databases, so that using phylogenetic algorithms we can predict how these sequences were sequences that actually are not existing in our planet anymore because they belong to extincted organisms. So we can predict, we can make reliable predictions, reliable proxies of these sequences. We can order those sequences to a company because right now we have facilities to synthesize DNA. So we just predict the sequences by phylogenetic algorithm. We write a Word file and we send to the company the sequence, the DNA sequence to the company, and they synthesize this DNA sequence. And afterwards we take this DNA sequence, we insert this DNA sequence into a modern host, in bacteria or in yeast, and we express those enzymes. This is so-called enzyme reconstruction and enzyme resurrection. Basically, we are predicting enzymes which were existing in our planet in the Precambrian or in the Carboniferous area, and somehow they are reliable proxies of the behavior in terms of biochemical properties of enzymes. All enzymes in our planet, right? So the idea here is whether we could actually resurrect an enzyme in the laboratory, and afterwards, by means of directed evolution, travel forward by adaptive evolution, so that we can recover one of these activities which are not in the modern counterparts. And these activities could be useful for many biotechnological applications. And also we could take the other way around. We could, from a modern enzyme, travel back by a technique of directed evolution, which is so-called genetic drift, in order to accumulate ancestral mutations and afterwards travel forward by adaptive evolution to achieve the same goal. So this concept of travel back and forth along the temporal line of evolution is going to give us a lot of opportunities in terms of engineering novel catalytic functions and more robust biocatalysts, while exploring natural evolutionary principles in terms of protein robustness and evolvability. And we've done this with Rubisco. Rubisco is one of the most ancient enzymes found in our planet, and is involved in the fixation of CO2 in the biosphere. And we collaborated with Spencer Whitney, who is one of the pioneers in Rubisco engineering, and also with Jose Manuel Sanchez Ruiz, who is pioneering the concept of enzyme resurrection. And we resurrected some ancestral Rubiscos. We traveled back in time, and we resurrected several ancestral nodes as old as 3.2 billion years old, which basically means in the Precambrian era. And we evolved these enzymes along with the modern counterpart by means of different techniques of directed evolution, and we compared the evolution of the modern enzyme but also the evolution of the ancestral one. And we obtained interesting properties in terms of the tolerance of ancestral enzyme. This slide represents the classical landscape of a directed evolution experiment, where one is the activity of the parental type, and these are the clones exploring a round of directed evolution. You can see here that the modern Rubisco, under the same mutational loading, is much less stable than the Precambrian Rubisco. And this is basically because the Precambrian Rubisco is a virgin mold for directed evolution, because in principle has not been subjected to natural evolution, and therefore is going to be capable to tolerate higher mutational loadings than the modern counterpart. And also we found out some interesting properties in terms of the conditions that were supposed to exist in the Precambrian era around 3.2 billion years ago. And to finish my talk, I've chosen also another example of my laboratory for the application of enzymes in the pharma industry. Peroxygenases. These are right now our favorite enzyme in the laboratory. Peroxygenases were discovered by my colleague Martin Hofrister from the Technical University of Dresden in 2004. And since its discovery was considered by many as a chimeric protein between P450s and peroxidases, because share the characteristic one electron oxidation reaction of peroxidases, but also the characteristic properties in terms of organic synthesis of P450s. Actually, peroxygenases can perform with high selectivity carbon, hydrogen, oxyfunctionalizations of a great variety of organic compounds. But unlike P450s, which requires the presence of auxiliary proteins and redox cofactors, peroxygenases only need to work hydrogen peroxide, which is the final electron acceptor and also the main oxygen source. They are soluble and extracellular. And we've done a lot of work with this enzyme in the last few years. The repertoire of reactions that this enzyme can perform is awesome. They can do brominations, sulfoxidations, N-oxidations, aromatic hydroxylations, alkyl hydroxylations, epoxidations, ether cleavage, and this is, as you can imagine, essential for the pharma industry. Indeed, the pharma industry is really involved in developing new drugs, new pharma compounds, and for this industry, it's essential to have in a large amount the metabolites of these pharma compounds in order to perform pharmacokinetics and pharmacodynamic studies. And we have demonstrated that we can evolve this enzyme to accept not the natural substrate, but to accept pharma compounds and to mimic the behavior of the human liver, where all these pharma compounds are transforming the corresponding metabolites. And using propranolol, which is a commonly used beta blocker drug for the treatment of high blood pressure, control of heart rhythm, or the prevention of migraines, we were capable of evolving a peroxygenase to produce with high selectivity 5-hydroxypropranolol, which is the main metabolite which is produced after the administration of propranolol in human beings. And we did this by the hand of directed evolution. We took the structure of our enzyme and we made in this helix random mutagenesis and recombination. Only in this helix, this is one of your colleagues here asked about whether it's better using directed evolution or rational design. Here we combine both approaches. Actually, we made, let's say, structure-guided evolution. Okay, so we use the structure to randomly mutating specific segments of the enzyme, and afterwards we obtain a mutant whose catalytic efficiency was improved to orders of magnitude, and also we achieve total turnover numbers as high as 265,000, which are very close to the industrial standards. We made also some computational calculations and simulations to understand the behavior behind these improvements using quantum mechanics, molecular mechanics, protein energy, landscape exploration algorithms. This is quite useful to understand why these mutations exerted this kind of effects in the protein, but also sometimes we can anticipate the mutations by using this approach. Is something that we are doing right now, trying to reduce the exploration of the vast protein sequence space using computational algorithms. So to finish my talk, this is important because we were so excited with these results that we've decided to found a company based on our results with peroxygenases and also other enzymes from the Lignity Consortium. And this is let me introduce EvoEnzyme. Is our spin-off, which is going to be released by the end of December, hopefully. And we are going to focus our activities in the design of projects customized on directed evolution and also in the commercialization of some of our evolved enzymes. And just to finish, a few conclusions about laboratory evolution. So directed evolution, I've talked today only about directed evolution of enzymes, but you can also find in literature examples of directed evolution of metabolic pathways or even whole microorganisms. Okay, so you can find here in this slide directed evolution of genome shuffling for whole cells or for genetic circuits, etc. So the scope of applications of directed evolution is broader than I explained here today. And somehow we are witnessing a revolution in the way of making laboratory evolution, incorporating computational algorithms as I've just explained, using quantum mechanics, molecular mechanics simulation, molecular dynamics. Also, we can evolve enzymes from extremophile environments. Right now, we are also trying to evolve enzymes using this ancestral approach of reconstruction and resurrection. And definitely, directed evolution has opened in front of our very eyes a door towards a wonderland full of surprises and challenges by which we can work in non-natural environments, in non-natural chemistry, in the synthesis of new biomaterials, new biosensors, new therapies, or the degradation of xenobiotics, to name just a few examples. And somehow directed evolution is contributing to the establishment and development of new techniques, whereas also is exploiting the power of synthetic biology, metabolic engineering, and system biology, all of them framed by the biotechnological rainbow, which is formed by the white, red, blue, and green biotechnologies. So summarizing, Frances Arnold said in her, when she was awarded with the Millennium Prize a couple of years ago, that it is not time for exploitation, because she's really worried about the conditions of our planet, and she's trying to contribute with directed evolution to improve, the sustainability of our planet. And she said it's time for exploration. And right now I will also add, it's also time for celebration, because all the people involved in directed evolution cannot be more happy that Frances has been awarded with this Nobel Prize in Chemistry, which she truly deserved it. And that's basically all. I would like to thank all the people involved in my research, and during all these years, special thanks to my students who are working with highest enthusiasm and motivation during all these years. And yes, guys, just let's do more evolution. And that's all. Thank you very much for your attention.